Have you ever wondered why some medications are just incredibly hard to take? Or maybe why a drug that seems really promising in the lab, works perfectly there, just struggles to actually help people in the real world? A lot of the time, the core issue is this really persistent challenge in pharmaceuticals: drug solubility. Basically, if a medicine doesn't dissolve properly, your body just can't absorb it effectively. And that's exactly why today we're taking a deep dive into something, well, I think it's truly fascinating: cyclodextrins. You can kind of picture them as these tiny natural molecular helpers. They're quietly revolutionizing how drugs are formulated and delivered.
Precisely. Yeah, in this deep dive, we're going to uncover what cyclodextrins actually are, explore their really surprising journey from, a lab curiosity to a pharmaceutical powerhouse, and then we'll really dig into the clever ways scientists are optimizing their use, all to create more effective medicines for you.
Right. Our goal today is to cut through the scientific jargon, get you right up to speed on the science behind these molecular baskets. We're pulling the most important insights from recent research. Think of this as your shortcut to being exceptionally well-informed. So let's get started. Okay, to really appreciate the impact cyclodextrins have today, I think we have to start right at the beginning, and it's actually a pretty surprising discovery that kicks things off. Way back in 1891, a French scientist, Avivier, first described this sort of mysterious substance. He had isolated it from a bacterial digestive starch, and, well, he just called it cellulosine. Not very catchy yet.
No, not yet. And that's just the first piece of the puzzle, really. Years later, an Austrian microbiologist, Franz Schardinger, he picked up that thread. He managed to isolate two distinct crystalline compounds from a bacterial digest of potato starch. He named them adextrin and botdextrin. And crucially, Schardinger identified his beddextrin as Villiers' original cellulosine. So that's when these compounds officially started being recognized as cyclodextrins, or CDs, specifically ICD and BCD. Then the family grew again in 1935 when Freudenberg and Jacobi introduced us to Gus cyclodextrin, Gus CD.
Okay, so even after their basic structures were identified, it sounds like for decades, maybe from about 1935 right up to 1955, scientists like Freudenberg and Kramer were working with them. But our sources describe them as, expensive chemical oddities?
Exactly, oddities. They understood the fundamental chemistry, how cyclodextrins could form these complexes with other molecules, but producing them in large, pure quantities was just incredibly difficult and super costly.
Right. And that really put a chokehold on any kind of widespread industrial use, didn't it?
Absolutely. But what's truly transformative here, the real turning point, was how biotechnological advances in the 1970s completely changed their trajectory. Genetic engineering allowed scientists to isolate these highly specific enzymes called cyclodextrin glucosyl transferases, or CGTases for short. These enzymes were much more active, much more specific, and that meant they could efficiently produce highly pure ACD, BCD, and GCD. This is what transformed cyclodextrins from those, prohibitively expensive curiosities into affordable industrial excipients.
Excipients. So basically, inactive ingredients that help the main drug work better.
Precisely. They help the active drug perform. And to give you a sense of this monumental shift in cost, BCD, which once cost a staggering, $2,000 per kilogram back in 1970. Today, it costs around $5 per kilogram. It's incredible.
Wow, that's a massive drop.
Huge. And today, its annual production is close to 10,000 tons. It's just a remarkable evolution.
Yeah, you can see how that dramatic price drop and the increased availability really lit the fuse. It led to this rapid, almost exponential growth in publications and patents related to them. Worldwide, cyclodextrins quickly moved from being this niche scientific interest to finding widespread use, especially in pharmaceuticals. It's actually fascinating to track their adoption timeline. The very first CD-containing pharmaceutical product was Prostorman E, those were sublingual tablets, launched in Japan back in 1976.
Japan was early.
Yeah. Then Europe followed in 1988 with peroxicamaz CD, which was known as Brexin tablets. And then finally in 1997, the first U.S.-approved product arrived. That was a triconazole 2-hydroxypropyl TCD oral solution, or Sporanox.
Sporanox, you're right. I remember that.
And today, it's just incredible to think that 35 different drugs are marketed globally in formulations that rely on these CD complexes.
35, that's significant penetration into the market.
Absolutely. So with that history established, let's pivot now to the how. What exactly are these cyclodextrins, structurally speaking, and how do they perform this molecular magic to improve drugs?
Okay, so at their core, cyclodextrins are essentially wing-shaped sugar molecules. They're derived from starch. They're made up of a specific number of glucose units joined together. 6 units for AZD, 7 for ACD, and 8 for ACD, although larger ones do exist too. Their structure isn't a perfect cylinder, though. It's more like a distinctive three-dimensional, I think, truncated cone, or maybe a donut-like shape.
So donut shape, okay.
Yeah, and what makes them so special is this kind of duality they have. Their outer surface is hydrophilic, meaning it loves water, it readily interacts with water, that's thanks to all the hydroxyl groups on the outside, but their central cavity is relatively lipophilic, fat-loving. That's because it's lined with skeletal carbons and what are called ethereal oxygens.
Okay, so water living outside, fat living inside, like a little molecular basket.
Exactly. That's a perfect analogy. A basket with a water-friendly outside and a fat-attracting inside. And this unique structure is really the basis for their popularity as pharmaceutical solubilizers. They can form what we call non-covalent dynamic inclusion complexes in solution. So imagine a drug molecule that doesn't dissolve well in water. That's the guest. And the cyclodextrin is the host. The guest molecule slips right into that lipophilic central cavity.
So it tucks inside the donut hole?
Precisely.
Okay.
And this effectively camouflages the drug's undesirable physical and chemical properties, especially its poor water solubility.
And the effects of this camouflaging, they must be pretty profound then.
Oh, absolutely. This interaction dramatically increases the drug's apparent water solubility. But it's not just about dissolving more, it also significantly boosts the drug's dissolution rate, how fast it dissolves.
Right, faster dissolving.
And that, in turn, can dramatically improve its oral bioavailability, which just means how much of the drug actually gets absorbed into your bloodstream and can do its job.
Okay, and that's especially critical for certain types of drugs, isn't it? The ones that struggle with solubility.
Exactly. Particularly for drugs classified as BCS class 2 and 4. These are drugs that generally have low solubility, but might have good permeability, meaning they could get absorbed, if only they could dissolve properly first. CDs help them do that.
Makes sense.
And zooming out just a bit, it's important to realize cyclodextrins don't only form these neat little one-to-one inclusion complexes, they're actually quite versatile. They can also form non-inclusion complexes, they can create molecular aggregates, almost like tiny clusters, and they can even stabilize what we call supersaturated drug solutions.
Supersaturated, meaning more drug dissolved than should be possible.
Kind of, yeah. It helps maintain those high drug concentrations over time, which can be really useful. And beyond just solubility and bioavailability, these versatile tools can do other things too. They can reduce or even prevent gastrointestinal and ocular irritation from certain drugs. They can minimize unpleasant smells or tastes in medications. Think about bitter pills. They can prevent unwanted drug interactions and even convert oily or liquid drugs into more manageable solid powders.
Wow, okay. That's quite a toolkit for one type of molecule.
Yeah.
Truly a multi-talented helper in drug formulation. So we have these natural ones, OSD, ECD, and DCD. Are they all equally useful across the board? Or do scientists need to be mindful of their individual differences when they're actually formulating drugs?
That's a really excellent question, because while these natural cyclodextrins are powerful, they definitely have some limitations. Take BCD, for example, the 7-unit one. It actually has relatively limited aqueous solubility itself.
The cyclodextrin itself doesn't dissolve well.
Right, which can sometimes cause the drug CD complex, the thing you formed, to increase solubility to actually precipitate out of the solution.
Oh, that completely defeats the purpose then.
Exactly, it sort of defeats the object. And this poor solubility is thought to be due to some strong intramolecular hydrogen bonding within its crystal structure. It kind of holds itself together too tightly in water. So the challenge then becomes, okay, how do we overcome this inherent limitation?
And what's the answer?
The answer lies in creating derivatives, modifying them chemically. Scientists discovered that by chemically substituting even just a few of those hydroxyl groups on the natural cyclodextrins, even substituting them with lipophilic or fat-loving groups, counterintuitively, their aqueous solubility dramatically improves.
Interesting. So making them slightly less water-loving in parts actually makes the whole molecule more soluble.
It seems counterintuitive, but yes, that structural modification disrupts the crystal packing and enhances overall water solubility. This is why you often hear about derivatives like hydroxypropyl beta-cyclodextrin, or HPCD for short.
UZCD, right?
And sulfobutylleather beta-cyclodextrin, SBECD. These modified, more water-soluble versions are generally considered non-toxic at low to moderate oral and intravenous doses. HPCD, for instance, is much, much more soluble and also toxicologically more benign than natural VCD. And it's found in several marketed drugs, with intravenous dosing approved up to a pretty high level, 16 grams per day.
16 grams. Okay, that suggests a good safety profile. Which leads to the next question. Given how complex these molecules can be and the modifications, how do scientists ensure their safety once they're actually put into medicines for people?
That's obviously a critical consideration, safety first, always. Cyclodextrins are generally quite large molecules. Their molecular weight is roughly 1,000 to over 2,000 daltons, and they are hydrophilic overall, especially the derivatives. Now, this combination of size and water-loving nature means they aren't significantly absorbed intact from the gastrointestinal tract when you take them orally.
So they mostly pass through.
Largely, yes. Interestingly, your body's own natural digestive enzymes, like the amylases in your saliva or pancreas, they don't really break down natural ACD and BCD. However, your intestinal microflora, the bacteria living in your gut, they can ferment them.
Okay, the gut bacteria get involved.
They do. Now, specifically for safety, while BCD itself cannot be given intravenously, partly due to that low aqueous solubility we mentioned, and also because it can have potential kidney toxicity at high concentrations via that route.
Right, no IV for VCD.
Correct. But when taken orally, it's essentially non-toxic. In fact, it's so safe orally, it's a common food additive in some regions. And specific acceptable daily intake limits, or ADIs, have been established for each type based on extensive safety studies. They vary quite a bit. 1.4 grams for ACD, 0.35 grams for DCD, and a much higher 10 grams for XCD.
That's quite a range.
It reflects their different properties and metabolism. And while some modified cyclodextrins, like certain methylated mesiezes, have shown effects on red blood cells in lab tests, what's called in vitro hemolysis, it's really important to understand that this doesn't directly translate to toxicity in the human body in vivo. It's more a way scientists classify their potential to interact with cell membranes. It's a factor that's carefully considered by regulators.
Okay, so lab tests help understand potential interactions, but aren't the whole picture for human safety.
Exactly. The key takeaway for listeners, for you, is that regulatory bodies like the FDA in the U.S. have looked at this extensively. They've listed ACD, UCD, and OCD as generally regarded as safe, CRAS, for food use. And furthermore, those important derivatives, HPCD and SBECD, are on the FDA's list of inactive pharmaceutical ingredients. This confirms their accepted safety for use in drug formulations.
That's reassuring. Okay, this brings us nicely to where the science is headed now. It sounds like cyclodextrins are pretty amazing tools, but what are the ongoing challenges in really optimizing their use? And what new discoveries are helping drug developers push the boundaries even further to create even better therapies for you?
Well, what's fascinating here is that even with such a powerful tool, accurately evaluating how effective they really are in complex, real-world pharmaceutical formulations is. Well, it's quite tricky. A lot of the foundational studies on cyclodextrin complexes were historically done in very dilute, very simple, ideal aqueous solutions, just water, drug, and CD.
The kind of clean conditions you only get in a lab.
Exactly. Which is rarely the messy reality of an actual pharmaceutical product like a tablet or a syrup, which contains lots of other ingredients, other excipients. So this raises a really important question. How do we get truly accurate data for these real-world applications where the environment the drug and CD find themselves in is much more intricate?
Right, because the lab result might not match the real-world result.
Yeah. Precisely. And a key limitation historically has been in the traditional measurement methods. The most common one, it's called the phase solubility method, often made this big simplifying assumption. It assumed that one drug molecule always paired up perfectly with one cyclodextrin molecule in a neat 1-to-1 ratio.
The simple guest host picture.
Yes, but recent understanding reveals it's often much more complicated than that. Cyclodextrins frequently form multiple types of complexes, different ratios, like 1.2 or 2.1, and also non-inclusion complexes where the drug doesn't fully go inside, not just that simple 1.1. So for poorly soluble drugs especially, relying on that old 1.1 assumption can actually lead to misleading results, like incorrect stability constants for the complexes.
Okay, so the math might be wrong if the assumption is wrong.
Basically, yes. And to complicate things even further, those other common ingredients in formulations, things like preservatives or polymers used to control drug release, they can also interfere with these measurements, making it even harder to get truly accurate data on how well the CD is working.
So what does this all mean then for the developers who are trying to make effective drugs? Is there a more reliable way to measure how well cyclodextrins are actually performing in these complex real-world scenarios?
Yes, absolutely. Thankfully, there is. A more accurate and frankly more useful method for determining the true solubilizing efficiency of cyclodextrins is to calculate something called complexation efficiency, or CE. This value, CE, is derived from the slope of those phase solubility diagrams we mentioned, but the crucial difference is that it's independent of the drug's own intrinsic solubility.
Independent of the drug's starting solubility. How does that help?
It helps because it gives you a clearer picture of how effectively the cyclodextrin itself is pulling the drug into solution, regardless of how poorly soluble the drug was to begin with. It makes CE a far more reliable metric when you're investigating how different pharmaceutical excipients might be influencing drug solubilization by the CD, especially in those complex real-world formulation studies.
Okay, so CE gives a better read on the cyclodextrin's actual power in a formulation.
A much better read, yes.
And here's where it gets, I think, really exciting. I was reading about a recent study that revealed something quite remarkable. Using mixtures of natural cyclodextrins, specifically cyclodextrin and cyclodextrin together, can have these powerful synergistic effects that go way beyond what either one could achieve on its own.
Indeed. That study was quite eye-opening. When the researchers looked at the solubility of a common drug like dexamethasone, they found something really profound. While the individual cyclodextrins, BIF-CD or GAY-CD alone, would eventually reach an upper limit on how much drug they could solubilize, often because they start forming insoluble higher-order complexes like drug CD-CD clusters.
Right, they hit a ceiling.
Exactly. But using specific combinations of both BIF-CD and GAY-CD could significantly raise this solubility limit. For dexamethasone, this meant increasing the maximum solubility from around 8 millimolar with just one type, up to an impressive 17 millimolar with the right mixture. That's more than double.
Wow, more than double the drug dissolved just by using a mix.
A substantial improvement, yes. And the study observed two interesting things. Sometimes they saw simple additive effects, where the total solubilization was just the sum of what each CD could do individually, predictable. But they also found surprising synergistic effects, where the mixture dissolved much more drug than you'd predict just by adding the individual effects together. The mechanism wasn't fully clear, but the result was significantly higher solubility.
Synergy. The whole being greater than the sum of its parts.
Precisely. And this discovery, it really opens up entirely new avenues for formulation.
So what this really means is that intelligently blending these natural molecular baskets, these natural cyclodextrins, could potentially be a really powerful, maybe even more cost-effective way to get more medicine into solution. It could expand the possibilities for using these natural cyclodextrins, perhaps as alternatives to some of the more complex and often more expensive modified ones we talked about earlier.
That's certainly the hope, and an active area of research. It could lead to a whole new generation of therapies that are both accessible and highly effective for you.
It really feels like we've journeyed quite a distance today, from that accidental discovery of cellulosine over a century ago, right up to the cutting edge of pharmaceutical formulation happening now. What really started as just an academic curiosity, maybe an expensive chemical oddity, has truly become a cornerstone of modern medicine. It's allowing so many poorly soluble drugs to be transformed into genuinely life-changing therapies.
Absolutely. And the broader implication here, connecting this to the bigger picture, is that cyclodextrins are just a prime example of how understanding subtle molecular interactions, really getting into the nitty-gritty, from those guest-host inclusion complexes to the surprising synergistic effects when you mix them, how that deep understanding can profoundly impact drug development. Ultimately, this kind of science makes medications more effective, safer, and critically more accessible to you, the patient.
This deep dive really highlights the incredible, often unseen innovation that's constantly happening in the field of drug delivery. It makes you think. What stands out most to you about these tiny molecular helpers? Maybe consider how many medicines you or someone you know might rely on daily that are only made possible because of the quiet, efficient, clever work of cyclodextrins happening behind the scenes.